Well, thank you very much for that very kind introduction. It's my pleasure to be here today. No? What I'd like to do is give a little bit of background on what is the microbiome and how it's related to health and disease. So what I'd like to do is give an overview of what is the microbiome so that we're all on the same page. What is the evidence that diet can influence the microbiome? And conversely, how can the microbiome influence the response to dietary components? And interwoven throughout this is what is the relationship between diet, the microbiome, and disease risk. So what do I mean when we're talking about the microbiome? Well, we've actually all been considered to be superorganisms. And that is because we are a composite of many different species. In addition to our eukaryotic or mammalian or human cells, there are also bacterial, viral, archaeal, and fungal cells. And while there's a lot of discrepancy in the literature, it's agreed on that there are at least equal numbers of microbial cells as human cells, and some estimates guess that there might be even 10 times more microbial cells than human cells. When we talk about the microbiota, we're talking about all the microbes living in a particular region or niche. And the majority of these live in the gastrointestinal tract. So our gut microbiota are the microbes in our gastrointestinal tract. And that's about 100 trillion organisms. The microbiome is their collective genome, and in fact there's about 100 times as many genes as the human genome. You might hear the term metagenome, which is a combination of both the microbial genes and the human genes, or metagenomics, which is a study of their function. So microbes have many important functions. They provide the ability to harvest nutrients. They produce additional energy otherwise inaccessible to the host. And this is mainly from the fermentation of soluble fiber to short-chain fatty acids. They can produce vitamins such as folate, biotin, and vitamin K. They can metabolize carcinogens like the heterocyclic amines formed in food during cooking. They can prevent colonization by pathogens, and they can assist in the development of a mature immune system. So about 10 years ago, NIH started to realize that the microbiome might have a very important part in health and disease prevention, and they wanted to understand what this relationship might be. So they funded the Human Microbiome Project. And the interest in this first project was what microbes are actually in humans. Does this vary among different sites of the body? Does this vary among individuals? And does this vary in between health and disease? So the question they asked was, who's there? Or to put this another way, is there a core microbiome that's present in all individuals, and does this vary among body sites? And they did this by funding two main projects. The first was a clinical healthy cohort in which they took 300 healthy male and female subjects, 18 to 40 years, and they sampled 5 different body sites. They collected 15 samples from men and 18 samples from women, and they collected samples up to 3 times in a number of year period. And part of the considering the people healthy was that they couldn't be on any antibiotics, they couldn't be taking prebiotics or probiotics, and they couldn't be on any immunomodulators. The other part of the Human Microbiome Project was really to say, is the microbiome different between people with health and people with disease? And they did this by funding a series of demonstration projects. In fact, 10 demonstration projects were funded. And if I had the slides, you could see the 10 different conditions. But the idea was they were focusing on major areas that might be associated with disease and trying to compare healthy individuals with diseased individuals. So pretty much this are the results. And what they observed was that in healthy Americans, the microbial composition of each part of the body is unique. Each dot on this slide represents a sample from an individual, and the different colors represent the different body sites. And as you can see, the different body sites clump together regardless of individual. And in fact, they estimated that for each person, there are about 1,000 bacterial species and 2 million bacterial genes. And overall, for the 300 subjects, there were about 10,000 species and 8 million genes. But interesting is there was no specific species that was present in every individual. And in fact, what they found was that the genetic potential of each microbiome is less variable. So what they observed was that there was no core microbiome, but although all of us have the ability to ferment dietary fiber, it might actually be different species of bacteria that are carrying out that fermentation. And the question that we need to be asking is not which bacteria are there, but what is their functional capacity. And they did this by funding the second phase of the Human Microbiome Project, which they called the Integrative Human Microbiome. And what it was is asking the question is, what actually are the microbes doing? And they did this by looking at the biological properties of both the microbiome and the host, looking at the microbial composition and integrating many different omics technologies from both the microbiome and host in 3 longitudinal cohort studies. And the idea was to develop integrated data sets as a community resource. The 3 microbiome-associated conditions that have been studied are the dynamics of pregnancy and preterm birth, inflammatory bowel disease, and type 2 diabetes. Well, I kind of just gave a little bit of a background on what NIH has been doing to try and understand what is the relationship between the microbiome and health, but I realize this audience is probably much more interested in what is the relationship between diet and the microbiome. Well, the evidence that suggests that diet can shape gut microbial profile in humans comes from a number of different types of studies. There's observational studies, such as globally distinct populations that have very different microbial composition, which I'll show in the next slide, but also long-term food pattern consumption. And people may have heard the term enterotypes. And this is the idea that regardless of geographical area, that people that eat similar types of diet might have similar types of microbiomes. For example, individuals that eat a lot of protein and fat tend to have a lot more Bacteroides, as opposed to those that eat a lot more fiber tend to have a lot more Prevotella. There's also evidence from short-term dietary interventions, such as low versus high fiber diets, animal versus plant food sources, and I'll show an example of that shortly. So this slide shows one of the first studies showing global population differences. This was comparing the microbial composition in children in rural Africa from those from Europe. And those in Africa eat significantly less calories, but significantly more dietary fiber than those from Europe. And what you can find is the children from Africa have significantly more Bacteroidetes, shown in green, and significantly less firmicutes, shown in red. And the specific Bacteroidetes that are changed actually tend to be Prevotella and Xylanobacteria. And why it's important is these bacteria actually have the enzymes to be able to digest xylin and cellulose. So they're actually able to produce the energy from those sources of dietary fiber and allows them to maximize the energy they're getting from the food that they're consuming. There's also evidence to suggest from feeding studies that short-term diet can alter microbial activity and gene expression. This is data from a short-term feeding study done in Turnbau's lab, where people were fed either exclusively a plant-based or an animal-based diet. The plant-based diet's in green, the animal-based diet's in red. And this slide has a lot of information on it, but I'd just like to get you to concentrate on this area, where they're looking at the similarities and differences in the composition of the microbiota in different individuals over time on the 2 different diets. And what you can see is all the red tends to clump together and all the green tends to clump together. So that the microbial composition is more similar when they're eating the plant-based diet versus when they're eating the animal-based diet, suggesting that it's actually overwhelming genetic differences. I don't normally go from human studies to animal studies, but this was an animal study that was published after that human study. And it also does suggest that diet dominates host genotype in shaping the mouse microbiota. In this study, 5 inbred and more than 200 outbred mouse strains were either fed a low-fat, high-plant polysaccharide diet or a high-fat, high-sugar diet, and their microbial composition was analyzed. The results are shown here for the inbred mouse, and you can see all the high-fat, high-sugar animals clumped together and all the low-fat, high-fiber animals clumped together. And that's specifically because when they had the low-fat, high-fiber diet, they tend to have significantly more Bacteroidetes. But when they had the high-fat, high-sugar diet, they tend to have significantly more firmicutes. So how can you use diet to modify the gut microbiota? Well, you can give probiotics, which are foods or dietary supplements that contain live bacteria, usually bifidobacteria or lactobacillus. You can give prebiotics, which are non-digestible food ingredients, which selectively stimulate the growth of bacteria. Examples include inulin, other oligosaccharides, lactulose, and resistant starch. Synbiotics, which are a combination of a probiotic with a prebiotic, such that the probiotic can actually use the prebiotic as a food source. And other dietary factors, many of the polyphenols in tea, cocoa, and wine can actually be used by the bacteria. And actually, spices are usually antimicrobials, and that's why they've been used for food preservation for centuries. Well, I'm not going to talk at all about prebiotics or probiotics since they're the focus of a later presentation today, but I'll use one example demonstrating some of the work on polyphenols in food. And in this study, we fed pigs different levels of cocoa polyphenols, and we looked at the microbial composition in the feces and the distal colon. And we found that when animals got high levels of cocoa flavanols, there was increased bifidobacterium in the feces and increased lactobacillus in the colon. Well, it's fine to see a change in microbial composition, but you actually want to see a biological effect associated with this. And what we found is that cocoa powder consumption also decreased the expression of TNF-alpha and toll-like receptors 2, 4, and 9 gene expression in the intestinal tissues. And all of these genes are associated with inflammatory processes, suggesting that the cocoa polyphenols were having beneficial effects in terms of intestinal inflammation. Well, I've talked very quickly about how dietary components can influence the numbers and types of microbes, but we need to realize that this is a two-sided relationship and that the microbes can also influence the response to dietary components. In fact, bacteria can produce new compounds from a number of different food components. Some of the foods that are metabolized by gut bacteria and the microbial metabolites are shown here, but on the limit of time, I'm only going to focus on these top two. So what is the relationship between dietary fiber and cancer? Well, the World Cancer Research Fund and the American Institute of Cancer Research summarized the epidemiologic evidence and concluded that dietary fiber is associated with a decreased risk of colon cancer. In fact, they found that for each 10-gram increase of fiber per day, there was a 10% decreased risk of developing colon cancer. Dietary fibers are fermented by colonic bacteria to form short-chain fatty acids, acetate, propionate, and butyrate. And butyrate is the most widely studied and the preferred energy source of colonocytes. However, butyrate has differential effects in normal versus cancer cells. And that's shown on this slide. When you consume dietary fiber, insoluble fiber actually speeds colonic transit time so that you'll have less exposure of the colonocytes to carcinogens. The soluble fiber is fermented to short-chain fatty acids, and butyrate is the most important. In normal cells, butyrate is the main energy source, as shown here. So the cells use the butyrate to actually increase proliferation and increase differentiation, and that helps to avoid muscle atrophy. In contrast, when cells are cancerous, they undergo something called the Warburg effect, and this actually causes them to use glucose rather than butyrate as the preferred energy source. And because they're already using the glucose for energy, it means that the butyrate can affect other molecular targets in the cell. And the important molecular target that it affects is called histone deacetylase, and it actually inhibits the activity of this enzyme. And because of its ability to inhibit HDAC, is you actually have decreased proliferation and increased apoptosis. There's also evidence to suggest that dietary fiber can affect bacterial diversity. And this is one of the most important studies that was published in the last couple of years, in which Justin Sonnensberg lab showed that when animals were on a high fiber diet, there was a large amount of bacterial diversity. And as long as they were maintained on this high fiber diet, they maintained their microbial diversity. However, if they were switched to a low fiber diet, there was a decrease in diversity, and it was further reduced over generations. If they were switched from the low fiber diet to the high fiber diet, there was some recovery, but not recovery of all the bacterial species, suggesting that some of them were lost. Well, why is this study so important? One of the things that we learn, or if you compare the microbiome between healthy individuals and people with a disease, be it colon cancer, be it obesity, be it metabolic syndrome, be it IBD. One consistent thing that you see in all these diseases is a loss of microbial diversity. As nutritionists, many of us realize that we recommend a high fiber diet. We know that it's beneficial, but we don't always know all the mechanisms whereby it might be exerting its beneficial effects. And I think this study clearly shows that probably one of the important ways that dietary fiber is contributing to many of its health effects is by maintaining bacterial diversity. There's also evidence to suggest that when fiber is present or it's not present, it influences the specific bacteria that are there and what they actually use as energy sources. In this study, they took gnotobiotic mice. That's mice that do not have any intestinal microbiota, and they gave them a combination of 14 different human microbes from the 5 major phylum, and then fed them a diet that was either fiber-rich or fiber-deprived. And what they found was that when mice were fed the high fiber diet, they have a very high mucus layer, and they're protected from infection by Siderobacter rodentium. In contrast, when they're fed a fiber-deprived gut microbiota, it actually increases the expansion of specific bacteria that can degrade the mucus and use the mucus as an energy source, so that they start losing this bacteria, that they start losing the mucus layer, and that they're more susceptible to infection with other bacteria and colitis. Purified prebiotic fibers did not alleviate the degradation of the mucus layer, and that germ-free mice on either diet do have a thin mucus layer, but are protected from pathogen infection. Another dietary component that can be metabolized by the gut bacteria are the Ellagitannins. These include sanguine H6, which is in strawberries and raspberries, penduculin, which is in walnuts, and vescaglin, which is in aged oak wine. But what all of these have in similarity is that they can be hydrolyzed to form ellagic acid, and then the gut microflora reacts with the ellagic acid to form urolithins. And urolithins have been associated with a number of health benefits. But there's large individual differences in the production of ellagic acid. This human crossover study took 10 subjects and fed them different amounts of different foods that contained ellagic acid, and then looked at the excretion of urolithids. And what you can see is there's large variability in the excretion, showing that depending on what microbes you have, it influences your ability to produce these metabolites. Another important set of dietary components that have been associated with microbial metabolism in disease are dietary carnitine, which is in meat, and dietary phosphatidylcholine, which is in eggs and cheese. The choline and the carnitine can be metabolized by the gut microflora to form trimethylamine, which is metabolized in the liver to form trimethylamine oxide. And in fact, the formation of trimethylamine oxide has been associated with increased risk of heart disease, stroke, and death. And I'm not going to talk much more. I'll do one more slide on this, but Dr. Ray is going to talk more about this shortly. But one thing that I'd like to mention is it's very easy to look at a single nutrient in isolation and say, this nutrient has this effect on the microbiome, and that's the expected health benefits. But you know, we don't eat single nutrients. We eat whole diets with varied foods in it. And in this study, they looked at the relationship between dietary allicin, which is one of the sulfur compounds formed in garlic, and how it influences the formation of TMAO after exposure to carnitine. And in this study, mice were either fed a chow diet, a carnitine supplemented diet, carnitine plus allicin, or just the chow and allicin for 6 weeks, and then given a carnitine challenge. This is the data looking at the production of plasma TMAO. And what you can see is in the animals given the carnitine diet, shown in orange, there's a large amount of TMAO production. However, when animals were fed the carnitine and allicin, it actually inhibited the formation of TMAO, suggesting that we really need to be thinking about nutrient-nutrient interactions. Well, this is just one study, but there was a study published about a year ago with dietary resveratrol showing a very similar effect, that resveratrol actually inhibited the formation of TMAO. Well, many of you may have heard about the French paradox. The question is, how come people in France can drink a lot of red wine, eat a lot of meat, and have a lower incidence of cardiovascular disease? Perhaps it's the production of TMAO might partially explain that. Just to bring one of the other areas that will be discussed today, there is a relationship between the human microbiome and foodborne pathogens. Organisms living on and within food have the potential to be either friends or foes to the consumer. And in fact, the human microbiome may decrease in diversity due to the exposure to antibiotics, the consumption of a high-fat and high-sugar diet, and decreased consumption of dietary fiber. Decreased diversity can increase susceptibility to invasive foodborne pathogens such as Clostridium, Staphylococcus, and Listeria. These organisms have greater potential to colonize and outcompete the host-associated community during dysbiosis. And in fact, some bacteria that are normal constituents of the microbiome and are sold as probiotics have the ability to become pathogenic, such as E. coli. So I've kind of given you a very whirlwind overview, and I hope that I've at least convinced you that both the specific numbers and types of microbes, as well as specific dietary food components and the amount of energy consumed, can influence the microbial metabolites forms, and then this can influence disease risk. Well, the answer is how can we utilize this information to make individual type recommendations for what types of foods people should be consuming to influence their susceptibility to various diseases. And this idea was published about 2 years ago, where they really looked at the relationship between the microbiome and individuals' responses to different foods. In this study, they had a cohort of 800 different individuals, and they looked at their glycemic response to different foods, and they found that there was large variability. One person can eat a banana and have a large spike in glucose, but no effect with a cookie, and the opposite for another individual. And they said, this doesn't make sense. How do we start to understand this? So they collected information from these 800 subjects on their gut microbiome. They conducted blood tests, questionnaires, anthropometric analysis, etc. And they put all the information into a computer to say, can we develop an algorithm to start understanding how individuals will respond? They did a validation cohort in 100 participants, and then they did a dietary intervention in 26 of the subjects. And what they found was there was high interpersonal variability in the post-meal glucose observed in the 800-person cohort, that using the personal and the microbiome features enabled accurate glucose response prediction. And in fact, the prediction is accurate and superior to common practice in this independent cohort. And then when they did the short-term feeding study, they found that the short-term personalized dietary interventions did successfully lower post-meal glucose. The question is, can we use this information beyond just looking at increases in blood glucose concentrations? So when you go and have your lunch today, take a metagenomic view of your dinner plate. Instead of thinking of the traditional four food groups, think of all the different dietary components and what their microbial metabolites might be. The take-home messages are that microbiome research is an emerging area of science, and there are many research opportunities available. The microbiome is integral to human physiology, maintenance of health, and development of disease. And in fact, there's a two-sided relationship between diet and the microbiome. And the food industry needs to stay actively informed about advances in this field. But most important, realize that bacteria are your friends. At lunch, remember that you're not just feeding yourself, and that the food that you want to eat might be different than the food that your microbes want to eat. And I'll finish with this and take any questions.
Thank you. That's very fascinating. So my question is, it not only impacts the food industry. As a representative from somebody who's been in crop protection, it also impacts how we look at studies in another way, right? How do we currently evaluate? First of all, I'm in regulatory, so I'm kind of taking a different perspective here. Mm-hmm. So my question to you is, how is this going to impact how we currently evaluate products in a different way, not just food, other products, because we know these enzymes are there, they're breaking it down, what's going on?
Well, I'd actually like to throw that question. There are a lot of people in the audience from FDA, And FDA actually handles the regulatory side of things. NIH kind of handles the science side of things. So if there's someone from FDA that would rather take a shot at answering it, it really is outside of the purview of NIH. That's okay.
Does anyone want to comment on that? Yeah.
Thank you for a wonderful talk. So, no study really investigated the presence of a eukaryotic microbe, including fungi and some parasite and so on. Are you having any kind of plan to study that or expand
So
your
there are
study?
lots of people looking at the fungi and viruses and stuff in different organ sites and how that influences. It's a definite important area of research. In a 20-minute talk, you really can't go into everything, and so much more has been done with the bacteria as opposed to some of the other organisms, and a lot more is known. But there are a lot of scientists looking at that now. The virome has large implications for lung diseases. That's probably one of the best examples.
Thank you. I actually had a quick question. So it is well established that the diet changes the composition of the gut microbiome, but those are typically samples that are looked at in fecal material or cecal material. Mm-hmm. What is known about the impact of the diet on changing the composition of the mucosal-associated microbiota, organisms that have co-evolved with the host?
So the answer is it's difficult to answer because it's hard to do those studies in humans. And so many of the human studies do the fecal samples because it's what's easy to collect. There are animal studies where it's easier to collect from many different sites throughout the gastrointestinal tract and to collect the mucosal associated versus the ones in the lumen. And you can see differences, but it varies from study to study, and the question is, can these be accurately extrapolated to humans?
Okay, thank you very much. Out of the interest of time, I think we have to move on to the next speaker and please join me to thank Dr. Davis for a very nice talk. Thank you.